Let's welcome in the series of transcription. This is a second video. So in this video we will discuss about different types of RNA. The central dogma molecular biology tells us the DNA holds the instruction for making proteins while RNA helps carry out their instruction. Messenger RNA act as a messenger, delivering the protein making instructions from DNA to the ribosomes, which are like protein making factors in the cell. Transfer RNA bring the building blocks of protein called amino acid to the ribosomes. Meanwhile, the ribosomes are mostly made up of another types of RNA called ribosomal RNA. Over the past 50 years, scientists have discovered the RNA does more than just help make proteins. Some types of RNA can actually speed up chemical reaction, similar to how enzymes work. Others have important role in controlling cell process. This means RNA many jobs in the cell, not just in making proteins. RNA that doesn't make proteins are called non-coding RNA. The abundance and variety of non-coding RNA, along with their important roles in controlling cell function, have led scientists to consider the idea that can RNA world have existed before DNA and proteins involved. What is non-coding RNA? Non-coding RNA is a type of RNA molecules that serve various functions in the cell, doesn't get translated into protein like typical messenger RNA does. These non-coding RNA are transcribed from a specific DNA sequence known as RNA genes. In more complex organisms like us, there's a diverse range of non-coding RNA, each with its own role. Some example of non-coding RNA includes familiar ones like transfer RNA and ribosomal RNA, which are crucial for protein synthesis. There are also smaller ones like microRNA and small interfering RNA that regulate genes expression. Additionally, there are longer non-coding RNA like exist and hot air that play roles in gene regulation and other cellular process. Altogether, these non-coding RNA contribute significantly to the complex machinery of the cell, despite non-directly building proteins. So now we need to understand, and let's give an example of non-coding RNA in eukaryotes. Eukaryotic cells boost a range of non-coding RNA beyond the commonly known ribosomal RNA and transfer RNA. These RNA play a crucial role in various cellular functions, although the full extent of their contribution is still being uncovered. Collectively, terms small regulated RNA, these molecules in eukaryotes are divided into several subcategories. From better understanding, the effect of these regulatory RNA are achieved through a mix of mechanisms, including matching complementary base pair, forming complex with proteins, and carrying out enzymatic activity. And we discuss about a small nuclear RNA. They are crucial for gene regulation through a process called RNA splicing. Found in the nucleus, the key players here are U1, U2, U5 and U4 and U6 particles, which aid in a splicing pre-mRNA to create mature messenger RNA. A small nucleolar within the nucleus of eukaryotic cell lies nucleolus, where ribosomal assembly and ribosomal RNA processing happen. A small nucleolar RNA were initially discovered in nucleolar extract due to their evidence in this area. The primary job is this process ribosomal RNA molecule, which involves modifying a specific nucleoside through methylation and pseudouridylation. There are two main types of a small nuclear RNA, CD box and HACA box family. The CD box usually add methyl group, while the HXX box wants isomerization uridine in the immature ribosomal RNA molecule. And next we discuss about a small sagel body specific RNA. A small sagel body specific RNA are a special type of a small nucleolar RNA found in a cellular suborganelles called the causal body. These organelles play a crucial role in the creation of a small ribonucleoprotein. Small nuclear RNA have a unique function. They act as a guidance for modifying certain type of RNA transcription by RNA polymerase II. Specifically, the lysosomal RNA U1, U2, U4, U5 and U12. These modifications include methylation and pseudouridylation, which are important for the proper functioning of these RNAs. The first small nuclear RNA to be discovered was U85. It stands out because it combines features of two common small nuclear RNA types, the CD box and HACX box small nuclear RNA. This allows U85 to guide both pseudouridylation and sigma methylation, making it distinct from typical small nuclear RNA. While U85 is unique, not all small nuclear RNA are composite. CD and HACX box small nuclear RNA. In fact, most small nuclear RNA are structurally and functionally similar to regulate small nuclear RNA. Their primary direction modification to ribosomal RNA in the nucleus. Another part of the cell involves in protein production. Now we discuss about microRNA. MicroRNA are teeny RNA molecules, typically 20 to 26 nucleotide long, which act as a regulator in cell. Their discovery or initial understanding came from research on the teeny roundworm C-like structure, elegant, but they have since been found in the many other organisms, including flies, mice and humans. MicroRNA have a key function. They can block the expression of genes by interfering with translation, the process that turns messenger RNA into protein. Although microRNA mainly work by attaching to messenger RNA and preventing translation, there are exceptions to these rules. Beyond their rules in regulating gene expression, microRNA have emerged as crucial players in various diseases, notably cancer, suggesting their significance extends for beyond basic cellular process. A small interfering RNA are a type of small RNA molecules, typically 21 to 25 base pair long, with a role in gene expression and regulation. The function by hindering gene expression primarily by one of the strands of a double-strand small interfering RNA integrating into complex called RISC. Once incorporated into RISC, the RNA containing complex can block the transcription of a specific messenger RNA molecules that matches its RNA. Initially recognized for their involvement in RNA interfering, a small interfering RNA must have evolved as a defense mechanism against virus with double-strand RNA. A small interfering RNA originated from longer RNA transcription through a process akin to the formation of microRNA. Both types of RNA are processed by the enzymes dicer, while a small interfering RNA and microRNA are typically to operate through different depression mechanisms. There are instances where a small interfering RNA behave more like microRNA, blurring the lines between the two classes. Now we discuss about PV interfering RNA. PV interfering RNA are the most abundant class of a small non-coding RNA molecules found in animal cell. They team up with PV subfamily aerogenate proteins to form RNA proteins complex. These complexes play a crucial role in epigenetic and post-transcriptional slicing. Their main job is to quiet down transposable element and other unwanted genetic material that might cause trouble. However, they are not just transposable element slicer, they also have a hand in regulating other genetic elements in germline cell. PV RNA are primarily produced from splicing a specific location in the genome called transposon traps. These traps act like RNA-based immune system protecting against transposon exception and invasion. Unlike microRNA, PVRNA are larger, 26 to 31 nucleotide base pair versus 21 to 24 nucleotide. They don't have consistent sequence across species. They are more complex and they don't really on dicer for the information, at least in animal. What are examples of non-coding RNA in prokaryotes? Bacteria have a set of small regulatory RNA that play crucial role in various cellular processes. These bacterial small RNA are involved in tasks such as regulating virulence and managing transition in bacterial growth phase, like moving from activating growth in the stationary phase, which happen when bacteria face conditions like a shortage of nucleotide. One notable example of a bacterial 6S RNA, which is present in Escherichia coli and is also found in many other bacterial species, suggesting its importance in genetic regulation. Researchers have discovered the 6S RNA influences the activity of RNA polymerase, the molecules responsible for transcribing messenger RNA from DNA. Specifically, 65 RNA works by inhibiting the specific subunit of RNA polymerase that typically boosts transcription during active growth phase. By doing so, 65 RNA hampers the expression of genes that promote active growth and aids in transition cell into a stationary phase.